Today we are going to see about polymorphonuclear neutrophils. Introduction. Neutrophils are the type of white blood cell with the multi-lobed nuclei and stainable cytoplasmic granules. These are the most abundant granulocytes, occupying about 40 to 60% of the total granulocytes. Neutrophils are like all other blood cells are formed from the stem cells in the bone marrow. After differentiation in the bone marrow, neutrophils are released into the peripheral blood and circulate for about 7 to 10 hours before migrating into the tissue, where they have a lifespan of only a few days. They are highly motile, allowing them to move in and out of the cell and tissue during infection. Structure of neutrophils. Neutrophils are mostly circular in range from 12 to 15 μm in human. The average size is 8 μm. Their shape change into amoeboid once they are activated, so they can extend to invader. These are the smallest of all granulocytes with characteristic multi-lobed nucleus with 3 to 5 lobes. The neutrophils are two groups. They are segmented neutrophils, band neutrophils. Segmented neutrophils. These are the mature neutrophils with a segmented nucleus. It is common type of neutrophils in bloodstream, responding to infection by engulfing and destroying pathogens. Band neutrophils. These are the immature neutrophils with a band-shaped nucleus. They are typically present in small number in the bloodstream, but can increase during infection or inflammation to fight off pathogens. Neutrophils are at the front line. Attack during an immune response are considered a part of the innate immune system. So, the cytoplasm of neutrophils contain primary acidophilic granules and secondary granules. The primary granulocytes contain myeloperoxidase, some lysosome and cationic protein. The secondary granulocytes contain lactoferrin, lysozyme and betal binding protein. Other cytoplasmic granules like mitochondria, Golgi complex appear sparingly, and the endoplasmic reticulum is entirely absent. Absolute neutrophil count is the test to measure the number of neutrophils and other granulocytes. The number of neutrophils in the blood is differ from person to person based on the age and the environment. In terms of cell count, the adult contain 1500 to 8000 cells. In terms of percentage of the cell, adult contain 40 to 60% of neutrophil. When the level of neutrophil is less than 1500, it considered a low neutrophil level. It is called as neutropenia. The neutropenia contain three types. They are mild neutropenia, moderate neutropenia and severe neutropenia. When the level of neutropenia greater than 8000, it is considered a high neutrophil. It is known as neutropenia. Structure of neutrophils diagram. Process of phagocytosis. Phagocytosis is a process where a certain cell like white blood cell engulf and digest the foreign particles, bacteria or dead cells. It is a crucial part of immune response to remove harmful substance from the body. The process of phagocytosis include following stages: chemotaxis, adherence and opsonization, injection, digestion include respiratory burst. Hemotaxis involves the movement of phagocyte to the site of infection, inflammation in response to hemotactic factor produced by the foreign particles or damaged or tissue. The various molecules like internuclein 8, hemokinase, C5a, C3b and the product of pathogen, formalin peptide. Adherence and opsonization. Adherence and opsonization are the crucial step in phagocytosis that enhances the ability of phagocyte to recognize, attach to and ultimately injects the pathogens and other particles. Adherence. In adherence process attached to the surface of target particles. Opsonization. Opsonization refers to the process of coating of bacteria by opsonin. Opsonin coated bacteria get attached to the surface of the phagocyte through the opsonin receptor. The principal opsonin are IgG opsonin and C3b opsonin. Injection. The adherent particle may initiate injection by activating an actin and myosin contracting system, which extends pseudopodia around the particle. The particle is evenly enclosed completely in a vacuole, the phagosomes. Lysosome granules come into contact and finally fuse with the phagosome, forming a phagolysosome. Several hydrolytic enzymes are now released into the phagolysosome, which act optimally at a low pH. Digestion. The respiratory burst is a rapid and intensive protection of reactive oxygen species by phagocytosis. During the process of phagocytosis, the burst of ROEs play a crucial role in the destruction of injected pathogens. It triggers the intracellular signaling event that activates the respiratory burst. It involves the binding of pathogens associated molecular pattern to pattern recognition receptor on surface of phagocytes. One of the central common for the respiratory burst is NADPH oxidase complex. The NADPH oxidase complex located in the phagocyte membrane and cytoplasmic granules. At the activation, it translocates to the phagosome membrane. The NADPH oxidase complex catalyzes the reduction of oxygen O2 to superoxidase anion. The process involves the transfer of electron from NADPH to oxygen. The superoxidase anion can further react to form a hydrogen peroxide or the superoxide anion to convert to hydrogen peroxide by the enzyme peroxidase dismutase. The hydrogen peroxide is react with the chloride ion in the presence of myeloperoxidase to form a hypochlorous acid. Finally, the killed organism and the cells are digested by the hydrolytic enzyme of the lysosome, followed by the elimination of the product of digestion to the exterior. Here it is the stage of phagocytosis diagram. The phagocyte can also kill neoplastic cell and the other cell directly without ingesting them, but just a membrane contact, which is known as contralateral cell injury. In addition, polymorphs having a cationic protein and a lactoferrin, which also have an antibacterial action. Killing by nitric acid. Nitric acid is formed within the most cell, particularly neutrophils and macrophages, generate a powerful antimicrobial effect. It is thought to be a particularly effective against salmonella. Thank you.